In the previous presentation, we have examined protein structure. So we looked at primary, secondary, tertiary, and quaternary structure of proteins. Now we are going to look at protein function as a consequence of its structure. So three-dimensional structure of protein is a consequence of primary structure of protein. It's a consequence of sequence of amino acids in a protein. And that has been shown in 1950s in studies carried out by Affinson. He carried out those studies on an enzyme. Of course, most of the enzymes, almost all of the enzymes are proteins. He looked at enzyme ribonuclease, and that's a single protein with single peptide chain with 124 amino acid residues. It does have 4 disulfide bonds, so it was very convenient for studies. Affinson used denaturing agents to destroy three-dimensional structure, tertiary structure of protein chain, and then investigated what conditions were required to restore this structure. As I mentioned, he used denaturing agent. Denaturing agents are agents that could be acids, bases, reducing or oxidizing agents. They do various things. They primarily interfere with intermolecular interactions, and so they destroy individual intermolecular interactions by forming their own intermolecular interactions with peptide bonds and side chains of proteins, and also they destroy disulfide bonds or break disulfide bonds. So they basically change three-dimensional structure of a protein without affecting primary structure of a protein. So they essentially destroy a natural three-dimensional structure of a protein without affecting primary structure of a protein. And here are some denaturing agents. Those would be agents that form strong hydrogen bonding and strong dipole-dipole interactions. Those are agents such as urea, chloridinium chloride, and then there is different type of reagent, that's beta-mercaptoethanol. That one cleaves disulfide bonds. So it cleaves disulfide bridges between different protein chains or within the same protein chain. In this case, in case of ribonuclease, there are 4 disulfide bridges within the same protein chain. Here you can see how mercaptoethanol operates. So we treat protein that contains disulfide bonds with excess of mercaptoethanol. And you can see a formula of mercaptoethanol. It's beta-mercaptoethanol or 2-mercaptoethanol. So it's ethanol molecule that has SH substituent on carbon 2 of the ethanol. When we use excess, it reacts with disulfide bonds within the protein and it forms its own disulfide bonds. It's simply an equilibrium process. We add very large excess of beta-mercaptoethanol, and so original disulfide bonds are cleaved and they are formed with mercapto group, SH group of ethanol. So that cleaves individual disulfide bonds in proteins, and that actually kind of untangles the chain. Chain is not really tangled, it has three-dimensional structure, but basically that destroys three-dimensional structure of the chain because chain, like chain of ribonuclease, is held together in particular globular structure by these disulfide bonds. So those 4 disulfide bonds are indicated here in different colors. And so they have to be in specific positions. For ribonuclease to be in its natural form and to be active as an enzyme. When ribonuclease is denatured, then chain is basically entangled, or chain assumes random unnatural conformation, basically just a random coil conformation. And in this case, by treatment with beta-mercaptoethanol and another denaturing agent that will break up intermolecular interactions, such as 8-molar urea, chain is completely denatured. It completely loses activity, and obviously, as you can see from this scheme, is in the form of random coil. Now, if denaturing agents are removed, let's say by dialysis, and then denatured ribonuclease is allowed to oxidize in air. Oxidation restores disulfide bonds. Reduction, of course, would destroy them, would break them up. But oxidation oxidizes these individual SH mercapto groups to disulfide groups. So oxidation in air completely restores activity of the native ribonuclease, which means that upon oxidation in air, correct disulfide bonds are formed spontaneously. So as denaturing agents are removed, then intermolecular interactions operate in such a way that chain folds correctly and only correct disulfide bonds are formed. So those that really should pair up. And that's obviously not a random process because there is only one way in which these 8 cysteine residues could pair up to form 4 cysteine bonds. And there are 104 different wrong ways for them to pair up. So very low probability process spontaneously occurs. On the other hand, if we only remove beta-mercaptoethanol from the solution and allow denatured ribonuclease to reform double bonds upon oxidation in air in 8 molar urea in the presence of denaturing agent that still interferes with intermolecular interactions with hydrogen bonding, then random wrong disulfide bonds form and so-called scrambled ribonuclease forms. That scrambled ribonuclease effectively has no activity because its chain is not properly folded chain and simply it's not an active enzyme. But if trace of mercaptoethanol, if urea is then removed and trace of mercaptoethanol is added, then scrambled ribonuclease will eventually restore its activity and will form or will fold into appropriate native form. And that's because trace of mercaptoethanol acts as a catalyst and breaks up individual disulfide bonds, and that allows correct disulfide bonds to form as wrong bonds have been broken and now correct bonds form. So from these experiments by Affinson, we can conclude that amino acid sequence specifies configuration. Based on these experiments, it is clear that primary structure of amino acid, sequence of amino acids specifies configuration. That means three-dimensional structure. That means secondary, tertiary, and in some cases, those with more than one chain, quaternary configuration, quaternary structure of amino acids. Native disulfide pairings drive the process of folding of the protein chain, correct process of folding. And in some proteins, refolding is not efficient, and protein molecules may tangle with one another to form aggregates. So in such cells, chaperone proteins block such interactions and help correct protein folding. And in fact, we'll cover role of chaperone proteins much later in the course when we cover biosynthesis of proteins. So we should look at how protein folding operates. And the process of protein folding is actually all or none process. So a process of protein folding is such that protein is either folded correctly. When we consider folded protein, it means folded in correct three-dimensional structure where the protein is active. So that will be natural configuration of a protein. Or unfolded, that will be denatured protein. So a process is actually all or none process. And there is sharp transition. So if there is unfolded protein, it very quickly folds into a completely folded protein. Or if protein is being denatured by use of denaturing agent, it very quickly unfolds into denatured protein. And when we look into process of folding, if we define 50% unfolded protein solution in which we have 50% unfolded protein, that means that in 50% unfolded solution, half the molecules are fully folded and half are fully unfolded. And that's an indication that protein folding is all or nothing process, because there is no, in 50% unfolded doesn't mean that individual protein chain is half unfolded and half folded. No, it's either completely folded, 100% folded, or completely unfolded. So protein folding is all or none process, and when we are talking about percent unfolded, that's how many chains are completely unfolded. So 50% unfolded means half chains are completely unfolded, half chains are completely folded. Now we're going to examine mechanism of folding. So each amino acid can assume 3 different conformations, each amino acid residue. And we have covered that previously. That means that there are those 2 angles, C and psi angles, on alpha carbon of each amino acid residue. And in general, they can assume, so as a consequence of possibility of assuming different C and psi angles, each amino acid residue has basically 3 degrees of freedom, can assume 3 different possible conformations. And if a protein is composed of 100 amino acid residues, then total number of different conformations for such protein chain would be 3 to the power of 100, or 5 to the power of 10 to the 47th. So huge number. So there is huge number of different conformations that are available to even a relatively small protein. 100 amino acid residues is relatively small protein. And if we assume that it takes very short amount of time, basically amount of time of single bond rotation, it will be 10 to negative 13 seconds to convert one structure to another, then total search time for a protein to examine all possible conformations and then assume the most stable one would be 5 times to 10 to the 47th multiplied by 10 to the negative 13 seconds, or 5 times 10 to the 34th seconds, or in terms of years, 1.6 times 10 to the power of 27 years. Now, you may be aware of it, that currently accepted age of the universe is 13.81 times 10 to the 9th years, 13.81 billion years. And that number changes a little bit as astronomers get more accurate, but you can see that it's much, much shorter than time needed to correctly fold one single small protein at random. So the entire age of the universe is not long enough for a single small protein chain to fold itself correctly doing a random search, which means that folding of a protein cannot be a random process. And that's actually this fact that protein folding is so quick, it occurs typically within milliseconds, and random process would take huge amount of time, longer than the time of the universe, is called Levinthal's paradox. We have different mechanisms to explain Levinthal's paradox. One of them is cumulative selection, which means that instead of completely random search through all possible conformations, partially correct intermediates are retained. So as soon as small segments of protein find correct conformation, those are retained. They don't search anymore through any further conformations, and that makes the entire process considerably faster. Another model is nucleation condensation model, and according to that model, interactions that lead to cooperative folding stabilize intermediates as structure builds up. So as local regions or secondary structures adopt their folded structures, they interact with one another, and that leads to progressively increasing stabilization and basically faster and faster folding. Now, another question is, can we predict a secondary structure from sequence? because if sequence of amino acids primary structure specifies secondary and then in turn tertiary structure, can we predict that? Well, actually amino acid sequence determines only about 60 or 70% of the secondary structure, while remaining 30 or 40% of the structure are determined by long-range intermolecular interactions. Long range, that means interactions of amino acid residues that are far apart from each other. For example, you may recall that amino acid residues that are 4 residues apart engage in hydrogen bonding in an alpha helix. So that would be this normal, typical short-term interaction, short-range interaction. But long-range interaction would be interaction within distant parts of the chain. And those interactions are very difficult to predict, and they determine about 30 to 40% of the structure of the protein, of the secondary and tertiary structure of the protein. And then, of course, we have cross-linking like disulfide bonds. There are 2 approaches to predict 3-dimensional structure from sequence. And the first approach is ab initio, which means from the beginning. And it relies on computer-based calculations of free energy of individual structure. So what is input is simply primary structure, and then a computer program simply calculates energies of each possible conformation based on steric hindrance on intermolecular interactions, and tries to come up with prediction of secondary structure. This approach has not been working very well, and there are a number of reasons for that. Simply our computers are not powerful enough as yet, even though they're very powerful. And reasons for that are that there is a very large number of possible conformations that the computer has to go through. So this is simply taxing computer time, and it takes a very long time for a computer to go through all the conformations. And then another problem is very small differences in stabilities between different conformations, because proteins have only marginal stability to start with, and very small change in conformation of a protein may not result in noticeable change in energy, and so computer may not notice any difference between different conformations. And then on top of that, weak intermolecular interactions, such as certain hydrogen bonds, certain dipole-dipole interactions, have very low energies, and that makes any meaningful prediction very difficult. So for that reason, at this time, knowledge-based methods are much better and have been much more successful. So we have large databases of known 3-dimensional structures of various proteins. And then we match amino acid sequence of unknown protein, of unknown structure, with those with known structures or fragments. And then if there is a match, then we predict that they would have same or similar secondary and tertiary structure. As I said, in general, this model has been far more successful compared to ab initio model. Now, while correct protein folding is necessary for their function, sometimes protein chains get misfolded, and that may result in various disorders, and some neurological diseases are consequence of incorrect protein folding. They may be caused by what we call prions, and such disorders are called amyloidosis. Misfolded proteins sometimes tend to aggregate, and so common feature of these disorders, amyloidosis, is a formation of amyloid plaques, amyloid fibers or amyloid plaques. Basically, proteins that are typically normally soluble proteins, which means that they are composed mainly of alpha helices, are converted into insoluble fibrils, and those insoluble fibrils are composed mainly of beta-pleated sheets. So those are called amyloid fibrils or amyloid plaques, and hence name amyloidosis for these diseases. So until recently, it was believed that diseases or infections are transmitted either by viruses or bacteria. But it turned out that for certain disorders, such as bovine spongiform encephalopathy, BSC, or mad cow disease, Creutzfeldt-Jakob or Jakob disease, CJD, scrapie, And actually some non-infectious diseases, such as Alzheimer's, Parkinson's, Huntington's, that actually amyloid plaques are present, amyloid fibrils are present. In case of infectious diseases, it appears that proteins known as prions, that are normally soluble proteins, they're sometimes labeled as PRP proteins, so they're normally soluble cellular proteins, and they're normally present in the brain and have their own function, are actually altered. They contain mainly alpha helices, maybe a few beta strands, but mainly alpha helices, but they are altered into PRPSC, PRP scrapie proteins, which are found in basically infected brains. And infectious agent could be actually this altered, misfolded prion. So if somehow, mainly to consumption of infected meat, such agent gets into the brain, then it will cause other normal prion proteins to misfold and to aggregate. And then they form those fibrils or plaques, and the brain basically loses function, cells begin to die out. Now, we don't actually know how these aggregates cause cell death. One hypothesis is that amyloid plaques themselves are non-toxic, but that their aggregates damage cell membranes and that way destroy cells. Another hypothesis is that amyloid plaque prevents transport of nutrients. So, as I said, these first three diseases are infectious and are transmissible, and in this case, proteins, prions are responsible, altered, misfolded prions. And what is interesting is that normal ways of sterilization, the way we would destroy viruses and bacteria, don't destroy prions. Prions are exceptionally stable. At this time, there is no treatment for any disease caused by prions. There are some experimental treatments, but nothing has been done yet. Similar misfolding of proteins, as I said, has been involved with non-infectious diseases, those that are believed to be non-infectious, such as Alzheimer's, Parkinson's, and Huntington's. And here is this misfolded fibril, as you can see from that was identified as a cause of Alzheimer's disease. This one is from Chemical and Engineering News article, and you can see how beta strands have been, how these proteins, individual proteins, contain beta strands and how they orient themselves and form, as you can see, fibrils. And this completes this part of study of function of proteins. Next, we'll look at a couple of specific proteins, function of a couple of specific proteins, and those are myoglobin and hemoglobin.